So how does translation work? How do we go from nucleotide sequences in messenger RNA to amino acid sequences in protein? So before we go forward, I wanted to kind of explain the reason behind these different words that we use for different steps of the gene expression. So when we copy the information from DNA to RNA, we call it transcription. And when we think about the word transcription, if you think about it on a everyday basis, it just a transcription of a conversation is just taking that same words in the same language that you have and just writing it down. So when someone says read the transcript, there was a conversation and somebody took that conversation and put it down on paper. So really the language remains the same, the alphabet remains the same. And in this case, with respect to nucleic acids, the alphabet is nucleotides. You're taking a message in a form of nucleic acid and you're changing it to the form of a nucleic acid. So it's just transcribing something, just like everyday language. But then when we take the message from RNA and then transforming it into language of protein, we're taking something that is in the language of nucleic acids and we are turning it into a language of amino acids. This is a different language. You're changing you're changing the language from the alphabet of nucleotides to alphabet of amino acids. And just like everyday life, when you're changing the words, meaning of the words and the letters, then you are translating something. So that's why these two steps, going from DNA to RNA, is referred to as transcription, and going from RNA to protein is translation, because the language of nucleotides and the language of proteins are two completely different languages because their basis is different. So just like any effort to translate something, we always need a good dictionary. And life also has a dictionary which we call it the genetic code. So basically, the dictionary of life shows us how to take the language of nucleic acid and translate it into a language of amino acids. Now, the words that would relate to an amino acid in nucleotides are three-letter words. Imagine a language in which words are made of all three letters. So we call this a triplet codon. So codons are triplet, 1, 2, 3. And a nucleotide goes into each of these positions. That's a triplet codon. And each triplet corresponds to a particular amino acid. And we can see that here. So for example, U, this triplet codon refers to phenylalanine. Now, there are three different positions that we need to fill here. And for each of these, there are 4 possibilities. So we have a 4 possibility here, and here, and here. So 4 times 4 times 4 is 64. We have 64 possible words that we can make in here. Okay? So 64 possible codons, which we call these codons. Each of these 3-letter signals or words, we call them a codon. And each codon refers to a particular amino acid. But how many amino acids do we have in nature? We only have 20 amino acids in nature. So we have more codons that we really need. So how does nature deal with that? Well, in 2 ways. First of all, some of the codons are stop codons. They don't code for anything, and there are 3 of them. Here's the 3: UAA, UAG and UGA are stop codons. They don't code for anything. They just tell the translation machinery to stop adding amino acids. Okay, so that takes care of 3 of them, but still we have 61 left. Excuse me. Now, one of these codons also is the initiation codon, which also corresponds to an amino acid called methionine. So we got 3 stop codons, 1 initiation codon. And then, okay, so now we have 60. So how do we take care of the rest? Well, there's redundancy built into the genetic code. So as you can see here, phenylalanine can be encoded by 2 different codons. Leucine can be encoded by 1, 2, 3, 4, 5, 6 different codons. Serine can be encoded by 4 different codons. So we say 2 things that happens in the genetic code. One is that the genetic code is redundant. That means that for every amino acid, there's usually more than 1 codon. There are exceptions. So for methionine, we only have AUG. And then I think it's the tryptophan, which also we have 1. Let me see if I can find it here. For tryptophan, there's also 1 codon. So tryptophan and methionine are exceptions. But for every other amino acid that we have more than 1 codon. So the genetic code is redundant. But there's another interesting thing about the genetic code, and that it is universal. So what does that mean? That means doesn't matter which organism you go to, from the simplest, smallest bacteria to the most complex multicellular organism, this dictionary is the same. That's why we can take a gene. These days, through biotechnology, we can take a gene, let's say for insulin, from human cells and produce it in bacteria and produce the insulin at much reduced cost. So 2 things to remember about the genetic code. It is redundant and it is universal. Now, how do you read these codes? So this is a table, and the way it's usually organized is that the first letter is on the left-hand side, the second letter is on the top, and the third letter is on the right-hand side. So then if I was going to use the particular code, C, this would be C, the first letter, U would be the second letter. So CU, these are all CU, and then the last letter is shown on the last column, which would be one of these. Okay, so this is what I just said. The genetic code is redundant and universal. So just take a few minutes and try to convert this RNA sequence into an amino acid sequence. So then, what is the machinery required to make protein? You need obviously the messenger RNA. You need the ribosome. You've met ribosome before when we talked about the organelles components of the cell. The ribosome have a small subunit and a large subunit, but you also need another molecule called the transfer RNA. Now, what is the transfer RNA? Well, we just talked about, for translation, we talked about having a dictionary. But then every time you want to do a translation, somebody needs to know how to read that dictionary. You need a translator. So the transfer RNA is life's translator. Someone who's a translator understands the language, the meaning of the word in 2 different languages. And a translator, the transfer RNA is pretty much the same thing. Why? Because of its 2 ends. On its 3' end is a place where a particular amino acid is attached. So here there are enzymes that attach particular amino acids to these transfer RNAs. But how does it match the amino acid to a particular codon? Well, there is a sequence in here on this loop, which is called the anticodon. And this anticodon is complementary to a triplet sequence on messenger RNA. So basically, this transfer RNA matches a codon with the proper amino acid. It's a really much simpler representation in here. This is messenger RNA. Let's imagine it says UUU in here. That's the triplet codon. Which transfer RNA would be matching with it? Well, a transfer RNA that has an AAA triplet on its sequence in this loop. And obviously, this sequence is going to be matched with the proper amino acid in here. So that's how transfer RNA acts as a translator. So just for an exercise for yourself, write down what would be the anticodon sequence for each of these triplet codons. So these are all triplets. What would be, just as an exercise, what would be the anticodon for each of these? Okay, so just like transcription, translation also have 3 steps: initiation, elongation, and termination. So the translation initiation starts by an initiator transfer RNA, which we said is always methionine, binding to the small ribosomal subunit. Now, how does the ribosome find the messenger RNA? Remember that 5' G cap that we talked about? And also there are sequences in here, which is called the untranslated region. These sequences will allow these ribosomal subunit, small ribosomal subunit attached to this transfer RNA to find the messenger RNA. And then these complexes will bind to the messenger RNA, and they start scanning down the messenger RNA until they find the initiation codon. Okay? So first they bind, then they scan the messenger RNA. As soon as they find the initiation codon and the transfer RNA, initiator transfer RNA, hydrogen bonds with the AUG, then the large ribosomal subunit finds the rest of the complex, and then initiation is complete. So this is the initiation complex. So try to remember, see if you can list the components of the translation initiation complex. The next step in translation is translation elongation, but before we start talking about it, let's talk about these 3 regions in the large ribosomal subunit. So there's the 3 regions are called EPA, and each of them stand for something. A stands for arrival. That's because that is the site on ribosome when a new transfer RNA comes in. P stands for peptide, which is the place where the growing peptide is sitting. And E stands for exit. That's where a transfer RNA that has unloaded its growing peptide enters in order to leave the ribosome. So let's imagine there's this messenger RNA. The ribosome is sitting here. There is a growing peptide sitting at this triplet codon, and a new transfer RNA binds to the matching codon. Then what happens is that this growing peptide is transferred to the and covalently attaches to the amino acid that is attached to the transfer RNA in the A site. So the transfer RNA in the P site unloads its load to the transfer RNA in the A site. Now the transfer RNA in the P site is empty, and then it moves to the E site. And the ribosome also moves one step down the line on the RNA. So once this empty transfer RNA enters the E site, it leaves the ribosome, and then the next cycle starts. And here's how it goes. So this egg-shaped molecule is the ribosome, and then here's the termination. So sequence here, it starts at AUG, the transfer RNA comes in, and then as soon as you reach a stop codon here, UGA, you see that a termination factor comes in and releases the ribosome. There is no transfer RNA that matches the stop codon, but there is a release factor that comes in, recognizes this, and releases everything. So this GIF is a nice representation of what happens. You see that the growing peptide is transferred to the one that is sitting in the one that arrives. So as it arrives, the growing peptide is added to it.